This program is brought to you by Stanford University. Please visit us at stanford.edu.
Our next speaker is my co-director of the meeting, longtime friend and colleague, Dr. Charlie Taylor, who directs the Stanford Cardiovascular Biomechanics Laboratory and has had a long-standing interest in trying to model and understand how blood flow affects blood vessels, basically. And a number of the people in the audience are either Charlie's current or prior trainees at many different levels. And Charlie's going to try to synthesize about 10 years of work in a single 30-minute presentation. Charlie.
Great. Thanks very much, Ron. I want to acknowledge some of the collaborators on this project and also some of the students. And I'll actually point out in particular Andrea Les, who many of the slides, probably most of the slides that I have are based upon some of the work that she's done. Sean Shadden's work will be shown there as well, and also Alberto Figueroa. And also there's a number of posters from some of my students during the poster session as well. Okay, so just a little bit of background, on risk of abdominal aortic aneurysms and maybe some of the potential role of hemodynamic forces. Some of the early studies, this is Volmar's study looking at World War II amputees, showing that there was a higher incidence of abdominal aortic aneurysms versus control subjects, and Volmar's work was predictive of, for single-leg amputees, of which way the aneurysm would bow, depending upon which limb was amputated. Ron and I and some very bright students did some work looking at the role of spinal cord injury in increasing the incidence of abdominal aortic aneurysms, and this was published in Journal of Vascular Surgery 2 years ago, showing that patients with spinal cord injury had almost a threefold increase in risk of abdominal aortic aneurysms versus normal control subjects. And there's been quite a bit of work looking at the role of peripheral vascular disease on the risk of abdominal aortic aneurysms, and I'm sure there's probably many in this audience that know far more about this work than I do, but peripheral vascular disease being predictive of an increased incidence of abdominal aortic aneurysms. Some of the impetus of this work, and actually our SCORE grant, really emanated from some of the work that was done in Ron's lab, showing that with the elastase infusion model, that with a high flow, increasing flow through the aneurysms actually led to a slowing of the growth of the aneurysms in the rat as compared to the low flow group. And really this is a background for our SCORE project looking at a way of increasing flow in abdominal aortic aneurysms in humans, that being exercise. And I think Ron showed this overall schema and flow chart for the project, and the work that I'll be talking about here will be Project 3, where we're really trying to focus on the hemodynamic environment. And kind of a cartoon image, and one of my students who's really a terrific artist sketched this up for me, and a cartoon schematic is that exercise therapy might slow the growth of aneurysms, perhaps by influencing clot burden, perhaps by changing lower extremity vascular conditions, reducing lower extremity vascular resistance. But this, of course, remains to be proven. Our work is to look at and describe the hemodynamic conditions in patients with small abdominal aortic aneurysms. And this, the first part of the project is taking imaging techniques, magnetic resonance imaging techniques, doing image processing, and this is software that we've developed in my lab. It's called SimVascular, to take medical image data and build 3D computational models, and then also using software we've developed to actually do blood flow simulations at rest and during dynamic exercise conditions to actually describe the fluid mechanical hemodynamic environment, wall shear stress, pressure, wall dynamics ultimately. So if we think about what might be some of the mechanisms by which exercise may slow the growth of AAA, increased shear or unidirectionality of flow may influence inflammatory cell adhesion, expression of vasoprotective genes on the portion of the endothelium, for instance, that might still be intact. In order to understand that, we can quantify shear stress and oscillatory shear index. Increased flow may also increase mixing of the flow in the aneurysm, reduce areas of flow stagnation and perhaps lessen the exposure of the vessel wall to inflammatory cells. And in order to look at that, we'll look at how fluid mixing and turbulent kinetic energy is affected from rest to exercise. Changes in peripheral resistance may reduce pressure decay time and also wall strain. And to look at that, we'll use imaging techniques and describe how vessel wall motion might be affected in patients with abdominal aortic aneurysms. So the first part of this and the imaging sequences that we're doing involve magnetic resonance angiography, and this gives us a three-dimensional volumetric data source and a three-dimensional image data source that we can then use our software on and build a 3D model such as we might see here. We also extract, using phase contrast MRI techniques, we measure blood flow at different locations. And we can take the flow at the level of the supraciliac and describe the total amount of flow coming into the abdominal aorta, and then identify the flow coming into the infrarenal portion in order to develop appropriate boundary conditions to extract the appropriate amount of flow through the branches. Once we have the right amount of infrarenal flow coming through there, then we can predict all the flow characteristics from that. So again, phase contrast MRI. We'll measure brachial artery blood pressure, and there have been some studies correlating brachial artery blood pressure with blood pressure at the level of the diaphragm. We'll use that data, and then the wall motion will also be obtained with magnetic resonance imaging techniques. And I'll focus most of the attention on the anatomic physiologic quantification. These are some of the subjects that we've imaged so far, and what we see, these are all magnetic resonance angiography studies and maximum intensity projections, and what you notice is really a dramatically different phenotype of abdominal aortic aneurysms. A side note, and I find this part really fascinating, this subject with the highly long tortuous iliac arteries was an avid cyclist, and we've seen that in a number of other circumstances, very long tortuous and large iliac vessels. Just kind of an interesting side note. But the point is that the AAAs are very different from one subject to the next. So we get the phase contrast data. We measure flow, and if we look at supraciliac flow and infrarenal flow, then we'll segment the vessel lumen over the cardiac cycle and then extract the volumetric flow as a function of time. And with the magnetic resonance angiography data, we have a workflow of building the models from image data, building 3D geometric models, and ultimately generating fine element meshes that can be used for simulation. We can then compare the image data to the computer models that are constructed from the image data. So for simulation of blood flow, and this is one of the big challenges, is that once we've discretized our model into a fine element mesh, we have to apply appropriate boundary conditions. You know, the flow through the major vessels is almost, entirely governed by the resistance of the downstream vascular beds. So the amount of flow that's going to go through different parts of the abdominal aorta will be determined on what's downstream. So what we do is we have simple electrical analogs. These are called Windkessel models. Frank developed these concepts about 100 years ago to describe the relationship between pressure and flow in the circulatory system. So simple three-element Windkessel models are used, in the parameters, the hydraulic resistance and the compliance characteristics. The parameters are adjusted to match the available experimental data. So we adjust the parameters to get the right amount of flow coming into the infrarenal aorta and to match the measured blood pressure as well. So we can do that, as well as possible in actually matching the experimental data that we have. Then we have to model and describe three-dimensional flow of blood, and we solve equations, partial differential equations called the Navier-Stokes equations on these models, and the solution to these types of problems involves, we have a supercomputer on the second floor with 2,000 processors, and it might take a few hundred processors for a few days to run a patient-specific simulation. For those of you that do this type of work, we've proven mesh independence of our solutions. We run up to 25 million elements in order to prove that we have mesh independence of our flow quantities, shear stress, etc. And these are numerical methods we developed. So when we start from rest, then we have to think about how to make changes for exercise. Changes in heart rate, we're simulating a 50% increase in heart rate. Changes in infrarenal flow, and we've made these measurements to support this. Changes in vascular resistance, target pressures, etc. And I don't have time to go into too many of the details, but be happy, or Andrea could answer questions about this afterwards. So if we have different quantities, pressure and velocity, we get that from the Navier-Stokes equations. Shear stress is derived from this flow solution. Oscillatory shear index is derived from that as well. And then we can calculate a turbulent kinetic energy. And Roger and Dr. Petey talked a little bit more about this, about how to do this. And I'll describe it in just a little detail too. So this is flow conditions so the colors I'll show you, it's the speed, how fast blood is moving. And we're using a volume rendering technique to actually look inside of the blood vessel so we can see the flow it's semi-transparent. And red is a high velocity flow, blue is low. So we see the blood is slowing down as it comes into the abdominal aortic aneurysm for this patient. And then we can look at what happens during dynamic exercise, increased flow complexity. The rapid color changes from red to blue indicates very complex flow. And this is for one subject. And here's another subject at rest, in more of a bilobe tortuous aneurysm at rest, and then simulated exercise. We look at the complexity of flow in the aneurysm at rest and exercise, and we can start to describe hemodynamic characteristics of that. So the first thing is to see whether we've compared, if we've set up our problem right and compared well to literature data. So we can, this pressure waveform basically shows that we can match the systolic and diastolic pressure. We can adjust our parameters appropriately to get that. We're getting the right characteristics of renal blood flow. This is our results, and this is the available literature data. Renal blood flow has a high diastolic component, which is one of the factors in flow reversal in the infrarenal aorta, and we can match impedance spectra at rest and exercise. So we have the right types of characteristics. In adjusting our parameters, we have a target infrarenal flow, and we can match that mean flow very well, but we can also do pretty well in matching the waveforms over the cardiac cycle in terms of volumetric flow in the infrarenal aorta. Once we've done the flow solution, we've gotten the velocity data, we can calculate wall shear stress, and we can look at different subjects. So there's 6 different subjects at rest and exercise, and we see, again, low shear at rest conditions, elevated during exercise conditions, but very heterogeneous shear stress distribution, but all of it is elevated. We don't really see regions in the aneurysms that are not, I would say, immune to the effects of exercise. Exercise really stimulates high flow throughout the entire aneurysm, and we can quantify the levels, the changes from rest to dynamic exercises at different locations, and get a handle on how much the exercise shear rate is going to be. We can calculate oscillatory shear index, which gives us an idea of the temporal reversal of flow over the cardiac cycle at rest and during exercise conditions. And really it tells us we see the differences are not as dramatic, but some of the oscillatory shear that is present at rest, is actually lowered in most locations with exercise. Quickly about turbulence. Is the flow turbulent? So we can take the total velocity that we've computed in this case, we can decompose it into a periodic portion, and then also the non-periodic portion, compute the kinetic energy of the cycle-to-cycle variation, and then plot that and see what it looks like. So we have turbulent kinetic energy at rest and during dynamic exercise. The first thing to note is that any evidence of turbulence is found in the aneurysm. It's not found, in the proximal section of the aorta during resting conditions. It is isolated to the aneurysm. And it's relatively low at rest conditions, and this will be dependent upon individual patient anatomy, but we see significant changes with exercise and significant increase in turbulent kinetic energy over the entire cardiac cycle during exercise conditions. This is not necessarily a bad thing, in my opinion, because what this leads to is a really significant flow mixing. It's still relatively, it would be a bad thing, in my opinion, if the shear rates did get high enough to, for instance, damage the endothelium in the areas where it's still intact, or if that led to rapid pressure fluctuations in the aneurysm. But my belief is that it's just increased flow and significantly increased mixing should have an impact on thrombus burden. Particle resonance time at rest conditions, and basically you seed the geometry, and this is work of my postdoctoral fellow Sean Shadden, who's really doing some phenomenal work in quantifying flow characteristics. So this is about 9.5 seconds to for dissipating and removing those particles from the aneurysmal region, and you see how that is reduced about threefold during exercise conditions. So again, the flow is mixing. We have flow stagnation during exercise. And here's some plots of the contour plots of the residence time throughout the aneurysmal region. So just some thoughts on this. We looked at the shear stress in the aneurysm, about 2 dynes per centimeter squared during rest, increased in all locations during exercise, eliminating any area of low wall shear. The oscillatory shear index decreased in all locations from rest to exercise. We have not done a large group of patients looking at turbulent kinetic energy, but we see in the subjects that we have done, it's essentially zero proximal to the AAA. At rest conditions, it's weakly turbulent in the aneurysm during rest conditions, but that increases dramatically with exercise. And the recirculation occurs, complex flow at rest and exercise, but the flow is still nonetheless well mixed, and exercise has a significant impact on residence time. I want to just make a few comments and actually show a few more results on some of the work that we're doing. It's ongoing work. These same patients, we're imaging them actually during dynamic exercise with an open MRI system that we have here at Stanford in an exercise bike that we built out of wood and plastic for this purpose. We've done a number of papers, we have a number of papers on measuring blood flow at rest or at exercise conditions using this setup, quantifying wall motion, and then also doing the simulations of fluid structure interaction, incorporating vessel wall motion into that. So the graded exercise, the rest, mild, and then more vigorous levels of exercise, we see here we can actually basically dial in the level of exercise and see how the hemodynamic conditions are affected by that to try to get an idea at what level of exercise, do we start to see some of the beneficial effects. On wall motion, I just want to share with you a few preliminary results just because they're so exciting. On the normal subjects versus the spinal cord injury patients, these are all patients that don't have abdominal aortic aneurysms. But what we see is that a reduced caliber of iliac arteries in these subjects. And the question is that, does this have any impact? We were measuring flow and wall motion, and we had some very interesting results that I'll share with you. The wall motion, again, with magnetic resonance techniques. So this is what we found, is that these are the spinal cord injury patients at the supraciliac level and then in the infrarenal level. And the difference in the circumferential strain was really dramatic. This is only 4 patients so far, but it's still, and the maximum strain is still statistically significantly different. And I was really struck by some of these differences between the control and the spinal cord injury patients, and there could be multiple explanations for this. But the fact that we see a much greater wall motion could be one of the explanations for the greater risk of abdominal aortic aneurysms. And I think maybe Jay might talk a little bit about this, about how mechanics can influence degradation of the extracellular matrix. Just a closing thought, there's been a lot of discussion about animal models, and one of my former doctoral students, Joan Grieve, who's here today, did some work for her PhD, and to be able to look at how shear stress changes with respect to body mass. And this, I remember the discussion I had with Ron, and when I saw some of the first data on flow, and he was computing shear stress from it, and I have to say I just didn't believe the numbers. I thought they were too high based upon our experience with flow in human aorta. And Joan developed a phase contrast MRI technique for our small magnet and measured blood velocity, calculated volumetric flow, and then looked at this question about how shear stress scales with respect to body mass. It's not invariant across species. Shear stress does indeed scale with body mass, and the scaling law that we observed experimentally, this wall shear stress scales with mass to the -0.38 power, was almost identical to that predicted by a paper by West's group in a paper in about a decade ago in Science. And now I think we understand why this is the case. But what you see, this is log of wall shear stress. So in a mouse, you see wall shear stress of 100 dynes per centimeter squared, and in a human, you're closer to 3, 4, 5 dynes per centimeter squared. There are remarkable differences in hemodynamics between small animal models and human aneurysms because of this scaling effect, because of the fact that Reynolds number is so much lower in a small animal than it is in humans. And this is quite an interesting observation. And I don't know yet what are the implications of this related to AAA models, but I would say without question that the hemodynamics are different. We undoubtedly won't see this type of turbulent or quasi-turbulent complex flow in a small animal model. Interestingly, and this is a work of another doctoral student of mine, Craig Gorshin, and he has a poster to talk about some of the other work that he's been doing, but circumferential strain is invariant with respect to body mass. So the amount of stretching over the cardiac cycle is invariant across species, but not so for the shear. And again, might be some interesting things to think about as we try to understand the relationship between animal models and human disease. At least the biomechanical aspects of that. I want to acknowledge some of the people that have been involved in the project, and a lot of it's more or less the whole SCORE team that's been involved in this. And I actually wanted to show you just one quick movie on the fluid structure interaction. You might find this interesting, but it doesn't seem to be playing right now. See if I can toggle it. Oh, it is playing there. Wasn't playing on my computer. I think I've probably ruined that. There it is. So basically the wall dynamics together with the pressure field and the velocity field and then wall shear stress over the cardiac cycle. And this is the computational work is work of Alberto Figueroa, and one of my grad students, Guangli Zhang, has developed new techniques for building models much faster than we could before. But it shows some of the wall dynamics work that we're trying to integrate into our future studies. So I thank you very much for your attention. So I'm moderating myself. If there, I'd be happy to take a couple of questions before Jay comes up. Yeah, Robert.
So if circumferential strain is invariant across species, I'm not sure what the question is. This might be a comment. That sort of implies that hoop stress is also invariant across species, which sort of implies that infrarenal blood pressure is invariant across species.
Well, it is invariant. Actually, blood pressure is invariant across species. It's I think one of the most remarkable things about the scaling. So it is invariant across species. Yeah. Yeah. Jonathan.
Thanks, Charlie. I just had a quick question regarding the Windkessel models, and you have beautiful reconstructions with a lot of bifurcations.
Yeah.
I wonder if you could comment on how much data there is out there to estimate those parameters accurately, and how that might affect things.
Well, it's a great question. And what we're doing, we're trying to, the challenge is that we measure flow at the supraciliac level and the infrarenal level. It's really not feasible to measure flow distribution through each of the branch vessels. It's just the data is just not, you don't have good enough resolution. So what we have to do is to be able to estimate the amount that's extracted. And it's largely based off literature data to be able to estimate the amount that's extracted. Once we have a target for flow distribution through, for instance, celiac vessel, SMA, renals, then we can hit that target pretty much exactly by appropriate tuning of the parameters. But I think the question, my belief is that it probably doesn't matter much how it's distributed through those proximal vessels. What matters most is how much flow is then coming down into the infrarenal aorta, which we're measuring. But we'll have to do a sensitivity study to prove that. I mean, we do know, as far as the characteristics of renal flow, I think we have a pretty good idea of what of that characteristic, but not the exact numbers. Yeah, David?
Thanks, Charlie. You got these very nice pictures with the fluid structure interaction. I know it's kind of apples and oranges, but can you compare the magnitude of the kind of effects on the calculated parameters relative to a non-compliant wall?
We haven't done that. We haven't done that study yet. I mean, we are measuring with MR. We're measuring wall motion at different locations, so we can then adjust our material parameters to match that measured wall motion to get the right level of compliance. I don't, I really don't know the answer to that as to what difference it's going to make. Other studies that have been done in maybe different vascular territories have indicated that there will be qualitatively maybe not as much of an effect, but there could be quantitative effect on wall shear stress by, it could be up to 50%. Maybe a little less in this patient group because their vessels are pretty stiff, relatively non-compliant. But I don't have a good answer for that. I mean, one of the main reasons for doing this, though, is for really understanding the dynamics in the wall and then to be able to transfer that to some of the growth and remodeling methods that Jay has developed and Alberto has talked about. I should point out, by the way, is that all of these simulations, it's the lumen. We're not including mural thrombus in there. For a rigid simulation, it doesn't matter. But we're not including the mural thrombus, and that will be, there are many challenges associated with that, and I know David will probably talk about the importance of a role of mural thrombus in AAA disease from a biomechanics perspective, but that's not part of the calculations right now. There's a lot of work to do, certainly, as you well know. Yep.
Charlie, I enjoyed your presentation. I have a question about your imaging techniques.
Yeah.
So when I look at these images, one of the best things for particle residence time would be the application of studying popliteal artery aneurysms that thrombose. And we don't know in small aneurysms, small popliteal aneurysms, when they're 2 centimeters, should we fix them when they're 3, especially in some of our older patients who have comorbid conditions? Is there a clinical practicality to looking at particle residence time in the popliteal aneurysms?
I don't know. I mean, I imagine there's probably a lot of clinical applications of being able to quantify residence time and flow stagnation. We haven't really explored those. A fellow sitting right in front of you is Sean Shadden, who's a postdoc that is developing those techniques. And one of the challenges, I think, will also then be correlating residence time with increased, thrombus burden. And that will be a big challenge to be able to do that. But it's clearly the direction that we need to go, is to say, what does it mean to have a factor of 3 greater residence time, from resting compared to exercise conditions? What does it mean, and what does it mean over the relatively brief period that one is exercising? Does it make a difference? I don't know the answer to that. So, Joe, you want to? Well, somebody should moderate me, I think.
We'll just keep asking.
But this is the last question, then
right,
Jay will come up. Yeah.
Okay, I enjoyed your talk. The issue of invariance across species I thought was really interesting, and you sort of mentioned the biomechanical aspects of it. Do you think also that could have some impact on the sort of biochemical aspects as well? Because you could imagine that changing Peckley numbers and a lot of the
it'll change transport characteristics, yeah.
As well.
Yeah, well, there certainly will be a relationship between any of the changes in fluid parameters or characteristics could certainly affect transport characteristics through the from the bloodstream into the vessel wall. I don't have an answer to that, but it would be, I think, quite an interesting thing to look at. So at the end message of that is that Ron was right about his experimental measurements. The theorists, I didn't believe it from a theoretical perspective, but anyways.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.